began with both a bachelor's and a master's of science in electrical engineering at MIT. He had a lot of fun in Cambridge, so he stayed there for the Harvard-MIT joint MD-PhD program, where he worked in Jim Fujimoto's lab during his PhD. After his training, he completed a residency in ophthalmology up the road in LA at USC Doheny. He further specialized in cornea refractive surgery at Emory. And in his illustrious career, he has been at many academic institutions, starting primarily in Ohio, at Cleveland Clinic, Ohio State, and Case Western. And then he was faculty at USC, where he was also in an endowed chair. Dr. Huang is best known as being one of the co-inventors of optical coherence tomography, which is, as you probably all know, a revolutionary technology that has influenced the care of so many people to this day. His work has led to numerous accolades, including the Lasker-DeBakey Clinical Medical Research Award. He has shaken the president's hand to receive the National Medal of Technology and Innovation. Sorry for the feedback. And he's received the Russ Prize for the National Academy of Engineering. With over 350 peer-reviewed publications, 64,000 citations, and 42 US patents, Dr. Huang's contributions to science and medicine are truly exceptional. Dr. Huang and I had a very busy, unusually busy afternoon, seeing 29 patients yesterday in clinic. All 29 patients received an OCT, and that guided their care in 29 instances. So thank you for the influence you have on all of our patients on a daily basis.
Okay. That's the same.
Well, it's an honor to be here. I remember being here probably in 2002 at a OCT conference of some type and meeting Zhongping, and he told me about Doppler OCT, and that actually got me interested in that area and eventually led to OCT and angiography, so very influential. That's also where I first heard about spectral domain being implemented. I think Brett Buma told me. Yeah. And I was here in 2020, just before COVID. It's like my last trip for a long time, and had very good, that memory had to last me for a long time. So I'd like to thank Dr. Chris Palczewski and Dr. Barry Cooperman for inviting me here again. I'm going to talk about seeing small and aiming big, the development and clinical impact of OCT. So it's going to be a bit of history, and I'll throw in some new stuff we're doing in the end. I do have financial interest in OCT and OCT angiography technology as listed on the title slide. So 2023, of course, was a big year for OCT, with the co-inventors receiving the Lasker Award and the National Medal of Technology and Innovation. I enjoy going to the White House very much. When the president tells you that you have literally changed the world for the better, you know, my head got like 2 sizes bigger. And I have to remind myself that a lot of other people contributed to the success of OCT, including Zhongping and I'm sure many others here. And the reason OCT was successful, I think, is not only because it gives us a micron resolution 3D imaging, but also because it's easy to use in ophthalmology. It's practical for translation, and that includes attributes like it uses invisible infrared light, so it doesn't dazzle the patient. It's much easier to use that way than the flash in photography. It only takes several seconds, and it's economical. We only get about $40 to scan both eyes. And it generates digital files that can be analyzed with computer and artificial intelligence these days. So it's very versatile. And I like that. And because of OCT success, I've been focusing basically my whole career on identifying technology that's translatable and bringing them to the clinic. And that's going to be it in my presentation. Of course, OCT give you micrometer resolution images in 3 dimensions. And you can look at that in cross-section or en face. And that's very useful in many diseases, including AMD and other very common eye diseases like diabetic retinopathy and glaucoma. And that's why it's used so much, because it's useful in managing the leading causes of blindness. I like to think of OCT as a combination of more than a century of research and development in using light to measure the time of flight of light, starting with Albert Michelson's invention of the interferometer to measure the speed of light, and of course, showing that it's independent of Earth motion, setting the stage for the theory of relativity more than a century ago. And we took the Michelson interferometer, the same design, and used that to measure the time of flight of light reflected from tissue to an equivalent of 30 femtoseconds, but in a much more economical and compact apparatus with better signal. And we all know that we can already measure depth in tissue using confocal imaging with confocal gating. The advantage of OCT is that coherence gating in most applications provide much better axial resolution, often by 1 or 2 orders of magnitude. And this is the beginning of OCT. Professor Fujimoto, who was my PhD thesis advisor, gave me a project to use an interferometer to measure the thickness of the cornea and retina specifically, but tissue in general. And this was a femtosecond laser lab that was setting records in the shortest femtosecond laser pulses generated. And these lasers are big apparatus that occupy most of the table. And so we cleared up like a small corner where I could develop an OCT system. And it's actually pretty easy to set up. And I was showing my father, who was visiting me then, of the experiments setup I was building. And so he knew I was doing something interesting and advanced at MIT, but he was wondering why I haven't graduated yet, because this was, I already been in medical school 4 years and my classmates are graduating and I'm just starting my PhD project. So it's like, I don't even know how many years I'll still be there. And, you know, I didn't have a very good answer. It's hard to justify the uncertainty, like, you know, I don't know when I'm going to graduate and I don't know how it's going to help my career. But, you know, in retrospect, I think it was worthwhile. And I was definitely enjoying myself then, doing something interesting, working with very smart people and learning from them and making rapid progress and having a lot of new ideas. And so one of the new ideas was OCT, and it started with measuring the thickness of the cornea and the depth of the anterior chamber, which was actually very easy to do. This is a project I finished in a few months. And I discovered that it's easy to obtain 10 micron axial resolution and more than 100 decibels of sensitivity. That means if you shine 100 decibel, it's 10 to the 10th. So 10 billion photons and only one of them come back, you still would be able to detect it, which is pretty amazing for a simple apparatus. And because of the high sensitivity, I was able to see signal from within the retina. So this was not what I expected. I thought like cornea, there would be like a strong front and back peak, and I'd be able to measure it. But usually there's a lot of peaks. And it's hard to say where the beginning and the end of the retina was. And so at first I thought, wow, this makes my experiments harder. It's a problem. And then I realized, oh, I can solve it by scanning the beam transversely to build up an image. Then I will be able to see by the pattern, you know, where the beginning and end of the retina is. And then I realized, well, if I can get an image of the retina, you know, down to the micrometer level, that's a big discovery and big advance. It's a new imaging modality. So that was kind of the aha moment for the invention of OCT. And we published the apparatus and initial results in Science in 1991. And that was also my PhD thesis. And I did not know at that time, but there were people in Vienna who were working on interferometry in the eye already, starting in the mid-1980s. But they took a different approach because their goal was to measure the axial eye length. So they use a Fabry-Perot interferometer. So these are two partially reflective plates in line. And when the axial eye length matches the delay between these two plates, they observe on an infrared scope this interference pattern, the circular pattern. So it's less sensitive, and they don't see the internal reflections from the retina. So it would be difficult to develop imaging with this. And that's how we were able to start much later and get to imaging much sooner. So we were fortunate. We started out with a different goal, and we had a different implementation. And OCT was possible at that juncture because of advances in optical technology, including semiconductor diode light sources, basically superluminescent diodes that have the spatial coherence of a laser, but the broad bandwidth of a LED. And then optical fibers, which provided single mode confinement to improve interferometry. And these were components developed for optical communications, the investment of many billions that we were taking advantage of. And these technologies were brought to OCT by Eric Swanson. I was very fortunate to be introduced to him by Professor Fujimoto. Eric was the leader of the MIT Lincoln Lab Inter-Satellite Communications project. And so he was an expert in building coherent receivers. And that is actually very similar to setup for OCT. So he was able to bring all the most advanced designs and components to this. And that's how we were able to progress very quickly. Eric also co-founded many companies in OCT and optical communications. The optical communications company like Sycamore and Acacia definitely built a lot more wealth for Eric and others, but I'm gratified that Eric thinks that his work in OCT was actually the one that made the biggest impact. And the patent that Eric, myself, Jun Fujimoto, and others filed in 1991 was the foundation of the first generation of ophthalmic OCT systems, and it included actually swept source as one of the implementations. So at that point, OCT was a solution in search of a problem. And we knew that it has an advantage of better spatial resolution, 10 or 100 times better than ultrasound, MRI, and computer tomography. So we knew we should focus on small tissue structures that are critical. And the two things we could think of were retina and coronary artery plaques. And that's our guess in 1991. We were fortunate that they were actually correct. By 2016, when we were asked to write a retrospective review, in the eye, there are about 30 million OCT procedures performed per year. And intravascular imaging was at about 100,000 procedures per year then. So these were the two biggest applications. And I also want to mention Michael He and Joe Izat, who helped develop the first retinal scanner for clinical studies. And I also want to mention the people who were responsible for the commercialization of OCT technology, which made it available to clinicians. And that included John Moore, who was the president of Humphrey Instrument and Zeiss, acquired Humphrey shortly thereafter. John actually thought OCT would be really great to complement visual field in glaucoma. Fortunately, he also funded some studies in retinal diseases. He had like 3 glaucoma clinics working on it and 1 retinal clinic. And that, of course, the applications in retina grew much faster. So it was a good thing he diversified. And without John, he supported this technology for quite a few years. I mean, by 2000, OCT still was not a commercial success, but he kept pushing for the 3rd generation. And that's really what made it made a lasting impact. So a good business partner is very important for the translational impact of a technology. As many of you know here, because many of you have successful startups that I see. And Jay Wei is the first engineer to implement OCT at Humphrey Zeiss, and he later founded OptiView, and I had the pleasure of working with him for several years. And these were the earliest OCT systems, OCT1 and OCT2, a very slow speed of 100 axial scans per second that produce grainy images. And only early adopters would want to use this because they're slow, there's a lot of motion artifacts, the pictures are grainy or smeared out. But that was enough. Some clinicians saw the potential in these grainy images because they could see some layers that they couldn't see on their direct examinations. It's enough to generate many publications and also a book in 1995 by Carmen Puliafito, Michael He, Joe Schumann, and Jim Fujimoto. And in 1999, when I was a fellow, a cornea fellow, I went to see a movie called Entrapment. I don't know if you remember it, if you're old enough, with Catherine Zeta-Jones and Sean Connery. And they were trying to break into some top secret vault in Kuala Lumpur. And there's a security scan, eye scanner. And, at first I thought, it would be an iris showing up on the screen, but no, they show an OCT image. And I was like, wow, wow, people knew about OCT. And then I realized there's a macular hole there. And like opaculated hole. It's like, why would he have a macular hole? It's like a glaring plot hole that makes no sense, but nobody else in the movie theater seems to be bothered by that. So what I learned early on was that translational research takes a lot of effort and perseverance. Clinical acceptance takes time. I mean, most retina specialists then thought it was just an evil plot so that residents would be relying on some, grainy technology and neglect to learn contact lens examination. But never mind those doubters. We keep improving the technology and clinical application. And good things will come. So for the next part, I want to take a longer historical perspective. 120 million years ago, a group of carnivorous wasps decided to go vegetarian and survive on pollens and nectar. And they happened to have color vision in both ultraviolet and visible wavelength. And because of that, that created evolutionary pressure for flowers to become colorful to attract bees so they can become pollinated. There's some kind of mutual evolutionary pressure. And together, they were very successful and spread throughout the terrestrial world. So being able to see something can change the world. It gave us this colorful world we have. And in analogy, early in the 2000s, OCT and anti-VEGF injections co-evolved. Because OCT was available, some clever retina specialists like Phil Rosenfeld is one I can think of, realized that when you see these probable neovascular membranes and retinal fluid in AMD, and you inject anti-vascular endothelial growth factor, which were the basic scientists discover were responsible for this disease, the fluid goes away and the vision can improve. And because this is a common cause of blindness, this saved a lot of vision. And it spread throughout ophthalmology practices. And so today there's about 2.5 million intravitreal anti-VEGF injections per year in the United States alone, and about 40 million OCT procedures. And they mutually drove each other's success. OCT guidance of as-needed anti-VEGF injection also saved money. It saves Medicare about $1 billion per year, which dwarfs the $0.4 billion investment in the first 20 years of OCT development by the federal government. So OCT became the most commonly used and cost-effective imaging modality in ophthalmology. By about 2002, reimbursement was less than photography and FA, and utilization is higher, and it kept climbing. In 2005, when I was actually moving from Cleveland to Los Angeles as a faculty, that year I went to AAO and there was an OCT image everywhere. And I thought, wow, we made it. It's an established part of ophthalmology. And so it's now used to detect and manage glaucoma, both in the anterior chamber looking for narrow angle and in the retina and optic nerve looking for optic atrophy. And we often can detect nerve fiber layer loss prior to visual field loss, and also monitor progression more closely. It's common now. And this took longer to get there. OCT became used more than visual field sometime in the 2010s. So John Moore's vision was eventually vindicated, but it took much longer. It's also used in cornea now. And one thing that I often use it for is to distinguish warpage, epithelial warpage versus keratoconus. In keratoconus, you have epithelial thinning at the same place as pachymetric thinning, and that's very, very specific. It can also do topography now. You can do topography with OCT because it's fast enough. And we have developed a topography algorithm and found it to be well confirmed, very comparable to other types of topography, from shine fluid camera to Placido disc. And there are, just last year, 3 systems that are FDA clear for anterior that have topography, including Heidelberg, CSO Italian company, and Visionics. So I think that's going to be used a lot. OCT also enable very precise surgery. This is the injection of gene therapy vectors under the retina. I know that's also an important topic of research here. And this is a surgery performed by Dr. Andy Lauer, who's the chair at KC. Very delicate surgery. Surgeons can have the manual dexterity to perform this. Of course, OCT gives you the depth perception necessary to do this reliably. You can also do this in the cornea. It's useful for separating the inner layers of the cornea in partial thickness corneal transplant. This is Anthony Kuo at Duke. So a lot of these new things, new advances in OCT are driven by improvement in the technology, specifically the increase in speed, which overall has doubled every 2.5 years since the original invention, kind of the Moore's law of commercial OCT speed. And that includes the spectral domain method, which uses a spectrometer in the detection arm, or swept source, which tunes laser at the source to give you the spectral interferogram that then you perform a Fourier transform digitally to get the axial scan. And of course, I first heard about spectral at a conference here. And this is basically parallel detection in different spectral channels. So the signal to noise advantage over the original time domain scales with the number of spectral channels divided by 2, which can be in the thousands. And again, this was made possible by components like high-speed line camera and tunable lasers developed for machine vision and, again, optical communications, and we took advantage of that. And so what I learned from that is keep my eyes peeled for new optical photonics technology if you want to advance OCT in a fundamental way. And these allowed us to extend structural OCT to 3D, to wider field of view or higher resolution. Also, even more interesting for us in research is novel contrast, OCT angiography, and optoretinography, which it's a new thing I would tell you about at the end. And of course, Doppler OCT as well. So higher speed allows us to do full 3D volumetric sampling of the retina and the cornea. And we can do that over ever wider field of view. And this, one of the impetus for this was pediatric examination in the neonatal intensive care unit for ROP. And there are many products in the market for retinal imaging. Some of them have pretty wide field, but none of them are OCT. So that motivated us to design a ultra wide field OCT scanner, handheld contact version that give us 140 degrees field of view, which is wider than the RedCam. And this is a video of Dr. Pete Campbell doing an examination in the NICU. He's very skilled at this, and it only takes a minute. And because the light is in the infrared, it doesn't dazzle the infant. They don't squirm as much as indirect ophthalmoscopy or RedCam, which use visible light. And this is the system developed by Dr. Yifan Jian with real-time display. So it's really gentler for the neonates and give you better contrast with a lot of features. You can see clear out to the ora serrata and ciliary body. You can see the ridge at the edge of the vascularized retina. It's also useful to evaluate tumors like retinoblastoma. You can measure the tumor volume or cross section. And again, it gives you a wider field of view than standard handheld OCT or photography. And you can see out in the periphery for retinal detachment. So I think ultra-wide field is an important area of research for us to look into pathologies of the peripheral retina. Most OCT systems on the market now look at the posterior pole, and this is a neglected area that could be important. Many blinding diseases occur there, including fibrovascular proliferation, neovascularization, retinal detachment, tumors, and so forth. And novel contrast is even more interesting for us. And in the past 10 years or so, a major focus of my research group have been OCT angiography led by Dr. Yali Zhao. And we developed projection resolve OCT angiography that can resolve 4 different plexuses in the retina. And this is now used clinically to look at diabetic retinopathy and AMD. So again, it's 3D, and you can see the normal retinal plexuses as well as neovascularization and choroidal flow. So again, retinal plexuses, neovascularization in AMD, and choroidal circulation. One of our most important contribution in this area is the development of something called split spectrum amplitude decorrelation angiography, or SADA. And that is based on what the problem is solved is that at the beginning, you have to capture many B frames, something like 8 or 10 B frames to compute the flow signal. And that takes too long to be practical. So we developed a method of capturing only 2 frames. And from each frame of OCT, you split the signal in the spectral dimension. And you can get as many as 11 spectral splits. And the spectral pattern is different because it's different in different colors or different part of the spectrum, and provide independent flow information. So you can compute the change in signal due to flow in these components and then recombine them. And it's like 11-fold increase in the number of images without increase in scan time. And this was demonstrated to improve the signal to noise ratio by a factor of 4. Also give you much better vascular detail. So now we can see 4 plexuses, the nerve fibular layer plexus and ganglion cell layer plexus in the superficial complex, and an intermediate and deep capillary plexus in the deep complex. And why do you need to know all this complex vascular anatomy? Well, it helps you diagnose disease. Optic nerve diseases typically affect the superficial complex. Outer retinal diseases affect the deep complex. And retinal vascular diseases like diabetic retinopathy affect all the plexuses. And neovascularization can be classified with some therapeutic implications. So this is an example of glaucoma, a common optic neuropathy. And it primarily affects the nerve fiber layer plexus. And if you focus on that, you can have high diagnostic accuracy. On the opposite end, retinitis pigmentosa mainly affects the photoreceptors and the deep capillary plexus. And diabetic retinopathy, of course, produce non-perfusion vascular anomalies in all three plexuses in the macula. And for CNV, we can see components below the RPE that's correspond to the occult component in FA, and above the RPE, you have something that's more like a classic finding in FA. And since 2014, when OCT angiography was first introduced as a clinical instrument commercially, there already been three generations in the past 10 years. The first versions have speed around 70 kilohertz or number of axial scans per second. And now we have 200 or even 400 kilohertz systems that provide better resolution or field of view. And of course, it's continuing to climb in Moore's law. And I'm very happy to report that in 2025, there will be a separate CPT code for OCT angiography, and it's going to pay 2 times regular OCT in terms of relative value units that tracks physician work and compensation, which is important. That's in recognition that medicine is also a commercial enterprise. In 2014, there's a first product launch of OCT angiography with OptiView. The first FDA clearance was actually with Zeiss. And then this led to early adoption by some clinicians, generated a lot of papers, but without better reimbursement, it's going to stay within a small circle. But now with its own CPT code and higher payment, I'm sure it'll drive a broad adoption. And that's very important. It's going to lead to incentive for the industry to improve the technology and adopt a lot of the advanced instrumentation and algorithms that we develop for licensing, so that this is actually pretty tremendous. It took 10 years for OCT angiography. Actually, for the original OCT, only took 5 years. So things are actually slowing down. The government is getting slower and slower. But it still grinds. So I think this will be a paradigm change in angiography. No injection compared to fluorescein or ICG. It's faster because you don't have to wait for dye transit. It's cheaper by far. And it's better in some ways, and better capillary contrast, for example, and better depth resolution. And it's getting better all the time. And because it's non-invasive, this is something you can use for screening and monitoring for quite a few diseases that affect the circulation. And so I predict it will be used a lot more than fluorescein angiography ever was. And it's, again, I think a big successful story for translational research. On the high resolution side, I'm excited about corneal microscopy. So we have developed a system using blue light OCT that actually can see cells really well, including endothelium and keratinocytes. And for some reason, at this wavelength, the collagen lamellae are really clear. And you can see microbes, fungi, and E. coli. And this is being packaged for clinical use. And I'm very excited that as a cornea specialist, that we might be able to see infectious agents in the clinic, which would be a first, and we're still working on that. Another novel contrast is optoretinography, and we have our own algorithm called against split spectrum SADOR, and I'll talk to you about that. This is a project led by Dr. Siyu Chen, an assistant professor at KCI. So intrinsic optical signal have been talked about for a long time. And even with fundus photography, you can see that the reflectance changes when you provide visual stimulus. And with adaptive optics scanning laser ophthalmoscope, you can see individual photoreceptors and their reflectance change when you flash light on it. With adaptive optics OCT, you can actually see photoreceptor outer segment length change. And the reflectance change and length change are linked. So basically the outer segment, I think of it as a Fabry-Perot interferometer. So there's interference between the reflection from the inner and outer terminations of the outer segment, and they interfere. So again, it's like a speckle and produce, so change in length produce change in reflectance. And that's different at different wavelengths. So again, that insight tells me that if you split the spectrum, you can enhance the signal. And again, we've shown that it does improve the signal to noise ratio. So by boosting the signal, that means we can use a larger spot diameter on the retina, covering multiple photoreceptors and still see enough signal to capture this photoreceptor activation. We use a ultra high resolution OCT system with 2-micron resolution, so we can easily resolve the outer segment. And then we flash visual stimulus and measures the response. And of course, our university development office loves putting the logo on it as visual stimulus. It works in inherited retinal degenerations. We're able to see changes, certainly in areas with abnormal microperimetry, but also we already see reduced SADOR response, even in areas with relatively normal microperimetry. And in dry AMD, intermediate AMD, we can see decreased response, especially in areas with drusen, large drusens, and subretinal drusenoid deposits. And this is in the fovea and parafovea. So I think it's good for early AMD, intermediate AMD. In late AMD, the signals are wiped out already, so it's hard to see. And we not only cone response, we can also see rod response by tracking the rod recovery after a intense flash. Dark adaptation is slower in patients with intermediate AMD. So now we can see a lot of things with OCT, not just thickness, edema, and atrophy as in standard structural OCT, but also we can see peripheral retina, we can see microbes, we can see neovascularization directly with OCT angiography. We can see ischemia by the capillary dropout in OCT angiography. I didn't talk about oximetry, but we can actually also do OCT oximetry. And now we can also see photoreceptor function. And when we can see something, we can affect it. And I'm pretty sure that before I retire, we'll be able to see therapies developed using these new biomarkers to gauge the treatment response. And I don't have to wait like bees for millions of years for the flowers to become colorful. I'm also excited about oculomics, or using the eye as a window to the brain, heart, and blood vessels. One thing we're working on is looking at Doppler OCT again, looking at the retinal artery waveform to assess systemic hemodynamics useful in carotid aortic valve diseases. And we found that in OCT angiography, especially around the fovea, we see different patterns of change in glaucoma, multiple sclerosis, and Alzheimer's dementia that might be useful in the evaluation of these systemic neurological diseases. And of course, OCT is useful beyond the eye. It's becoming fairly important in coronary artery disease to guide the placement of stents. There are some studies that show better results with OCT guidance. And you can also use it in the skin to look for cancer, inflammation, vascular malformations, in the digestive tract to look at cancer, look at vibration in the ear. You can look at the brain, you can look at the teeth. There are many tissues that used to be too big for OCT to tackle because our resolution is so fine. It's easy to generate gigabytes of data just over the retina, tens of millimeters. When you think of skin or GI tract, it's 1,000 times bigger organ. It's hard to tackle without a very high speed that we now have with OCT, up to megahertz. For Zhongping, probably know that very well, and I'm excited to see what you're working on. But there are a lot of potentials. So that wraps up my talk, and I want to acknowledge the support of NIH, RPB, Malcolm Marquis Endowment, and also a lot of companies that work with us. And this is our research group, the Center for Ophthalmic Optics and Lasers, or COOLAB. It comprised 6 faculty members and about 40 people in total. Please visit us at coolab.net. Thank you.
Thank you very much. We will start with questions from the room.
Hello, Dr. Huang. My name is Jacob Diaz. I'm a medical student actually studying optoretinography using photostress and the photoreceptor recovery. And I was wondering if you think the commercial OCT systems that are currently in clinics now have the power and the ability to assess this photostress recovery at a reliable rate in clinic, or is that more reserved for the higher acuity systems that you see in a lab setting?
Yes, I think the current commercial systems, if you outfit them with a proper visual stimuli, can already measure optoretinography with an efficient algorithm. But they're not optimal. I think in order for this to cover, to have reliable signal-to-noise ratio to cover a wide enough area, you would want something with better axial transverse resolution and higher speed. But we're actually not far away. I think in 10 years, I predict that optoretinography would be useful in the clinic.
Thank you.
Hi, Dr. Huang. I'm Cher from Cognitive Science. I do adaptive optics. And I have 2 stupid questions. The first question is, the traditional Michelson interferometer uses a laser. What were your thought processes going from a laser to the SLD? And now what are how does that compare to the swept source now coming back to a so-called a laser again?
I don't think it actually matters that much. And I don't think Albert Michelson had a laser because that was 18 something. So I think traditionally Michelson did not use a laser. And what is important is that you have mode matching between the reference and sample arm. And of course, using a I think the microphone is off something. So. We'll fix it. issues. So what's important is matching the transverse mode of the reference and sample arms. And the superluminescent diode, even though it's not a laser because the mode is confined in the semiconductor structure, actually can generate single mode beam. And we also usually implement OCT in a single mode optical fiber, which matches the mode. So either way, whether you use a laser or SLD, we are in single mode operation. There are also people doing multi-mode OCT at a small avant-garde group. It would be much more difficult to analyze than the usual OCT signal using single transverse mode, but that's also possible. And actually, you can recover more information about scattering.
Okay, thank you. The second question is, when you do your split spectrum, do you do swept source or do you use the regular SLD?
You can do either.
Okay.
You can do swept source or SLD.
Okay.
Thank you.
Let's go to web.
We actually don't have the question on the web yet, but let me ask my question. So you show out your vision for using the OCT for the development of new therapies. And in our research here, we use a lot of mice. So do you see that these latest advances could be used also with mice, or maybe mouse eye structure is not yielding itself to this, to applying this? I really love your.
Yeah, I think so, because it's hard to do microperimetry in mice. They don't respond that well. And if you can objectively measure their photoreceptor function, for example, that would be brand new information that you didn't have before on their retinal function, for example. It's just one thing.
We have one more question here, and then I'll go on the other side.
Hello, sir. My name is Atul. I'm postdoc under Dr. Brown. And I just want to ask that OCT has reached its upper limit, because when OCT was developed, the technology actually in micro scale, so it matches with the OCT technology. Right now, the current technology in nanoscale, that 6 nanometer. So the other technology can replace OCT right now, because OCT has a diffraction limit problem, and like in lateral resolution.
6 nanometer imaging with
No,
what?
the current semiconductor technology that is actually for spectrometers and that for detection is right now in 6 nanometer scale. So OCT has a diffraction limit problem. It can go maximum to 200 nanometer, but right now in your resolution in micron.
OCT local displacement.
Oh, yeah. I mean, yeah, of course, you can resolve it. I think you might be talking about resolving phase differences that correspond to a few nanometer. That's possible. Of course, you can do sub-wavelength measurements of phase, but that doesn't apply to most anatomic measurements. I think I know that in the ear, you can measure vibration down to that limit, for sure. I'm confused on the implications.
Well, I was going to follow up on the same in a certain sense. Great presentation, love hearing the history. And again, this technology has revolutionized eye care and retina care. So we're still looking forward to a future where we can get one more order of magnitude of resolution to get down to single cell photoreceptor measurement. What are your thoughts on what it'll take to get there?
There's a lot of people doing adaptive optics.
Right, that's been the pathways adaptive optics. Yeah.
I'm actually trying to run away from it because I think that's limit your field of view and make it difficult for clinical translation. And I'm mainly interested in clinical translation within the next 10 years or so. So boosting the signal without single cell resolution is my approach.
And on a separate but related, on OCT angiography, again, it measures more flow, but the fluorescent angiography can also show leakage. Thoughts about how you could, are there any chances of being able to show leakage in addition to flow with OCTA?
Well, some people has suggested to me if I can see the flow associated with leakage, but that is so slow, I don't think that's feasible. There are people who are simulating leakage using artificial intelligence based on structural and flow signal. And it can look very convincing, although I'm not sure that it's absolutely reliable. So I don't, maybe that would be a solution, but it's somewhat difficult to validate. But I like to leave, some diseases where leakage is most important, like vasculitis, where I think it was still continue to be useful for FA, so you can keep your photographers employed. I think that's a good thing.
Well, ultimately on OCTV, we're able to see the product of leakage by swelling.
Right.
It's different than looking on the OCTA.
Yeah, I think that's a signal that can be, used to backward predict the leakage on those artificial intelligence algorithms.
Congratulations on your award. And we are trying to look at what will be the next technology. And one of the things we are doing, and a few other groups doing, is look at elastogram.
Mm-hmm.
And obviously there's a application in anterior chamber, but we start to look at posterior chamber, look at the mechanical property of retinal, choroid, optic nerve, and but we are still trying to find a clinical problem that require those information. Can you comment on essentially how elasticity can impact clinical trial?
I will protect you from this.
Yeah, elastography is very, very interesting. And I know most people in that area are working on cornea because there's a disease called keratoconus where the cornea become more plastic and elastic. The modulus is decreasing. There are techniques to measure elastography, theoretically at least, could lead to earlier detection. And then in glaucoma, mechanical distortion of the lamina cribosa is theoretically, where the disease occurs. So there, maybe you're working on that as well. I don't know. I'm sure a lot of diseases affect retinal elastic properties, and it's pretty much unknown. I think so. I'm sure blazing a new trail if you're measuring the retina. Cataracts, the lens, of course, changes elasticity. Although I'm not sure that's super useful since we just take out the cataract anyway.
All right, let's go to Zoom.
Piotr Kasprzycki, do you have a question?
Yes, hello. Can you hear me well?
Yes.
Yes, I have a question. Actually, according to the polarization sensitive OCT, is there any place for clinical practice use of this method, or is it too hard to commercialize or translate? What is your opinion?
I know PSOCT is very powerful in providing contrast for retinal pigment epithelium. It's a good way to look at birefringent tissue like the cornea and other oriented connective tissues, collagen lamellae. But I haven't seen it commercialized, even though it's been developed to a pretty high degree of refinement over the past 20 years. I actually like the idea a lot because I provided the concept for the first paper for it at MIT. That was 30 years ago now, but I'm not working on it because I'm beginning to despair that it's maybe too complex for commercialization. Just empirically, actually don't think it's that complicated, but for some reason the companies are not picking it up. I don't know why.
Okay, and we have a comment and question from Dr. Wojtkowski,
Mhm.
and he asked that I read it. David, thank you for your talk. I am curious what your understanding of the optoretinography effect is. Can you say with certainty that the observed brightness changes correspond to photoreceptor activity?
I think there is evidence in that the strength of the signal scales with the brightness of the visual stimulus. So I would assume that it's from photoreceptor activation. And it, spatially, we have ultra high resolution OCT, so we are sure we're measuring the outer segment. I guess there could be alternative explanations, but I'm hard pressed to think of what else it could be. I'll be interested to hear Marche's thoughts.
Okay, thank you.
We have one more.
We have one more from the Zoom. Juemi Zhuo, are you there to ask the question?
Yeah, I can ask a question. Yeah. Hi, David. Thanks for your talk. Quick question regarding the ORG. Do you think there are refractive index changes which contribute to the total change in the OPL measured by phase-sensitive OCT, or just the path length change? Sorry, the physical length changes.
I think the mechanisms would make a physical length change more likely, even though because OCT just measures the optical delay, it really cannot distinguish between refractive index, group index change or physical length change. So I think it's mostly physical length change, but I don't have experimental evidence to prove that to you.
Got it. Cool.
I'm not particularly interested in tracking that down. I just know provides signal that's clinically relevant. I'm translation heavy.
Thanks.
We had a wonderful discussion, so we will finish with Dr. Brown.
Great, thank you so much, Dr. Huang. Really nice coverage of kind of the historical perspective of OCT as well as the future opportunities. Before your talk, we were discussing industry's adoption of new technologies and some risk aversion. So you showed us different techniques for angiography. You showed us wide field imaging. You showed us optoretinography, looking at function of the outer photoreceptors. If you were going to encourage industry on one thing that's low risk and likely to be useful, what would you advise?
I think they already voted for wide field because there's a lot of wide field product coming out of industry, especially from China. And certainly peripheral retinal diseases is so far neglected and clinically important area. And it's relatively straightforward. So I think that would be number one. And I'm more interested in optoretinography because it's new contrast. So in terms of research, getting NIH grants, that might be more plausible. I'm still in the grant-funded research business, even though I'm interested in startup companies too.
I think we will add one more question. Yes, the anxious student.
Pressure. Hi, Dr. Huang. My name is Emily. I'm a graduate student in Dorota's Grounds Graphics Lab. Can you talk a little bit about the implementation of artificial intelligence with OCT and how this may continue to synergize this technology? Thank you.
Artificial intelligence has become more and more important, and we're investing more and more in it. And its use now, especially in our OCT and geography projects, to process the signal to get rid of artifacts. So that's one thing. And then it's important for segmentation, more reliable than a traditional image processing, to distinguish between the different retinal layers and plexuses. It's used to recognize disease features like drusen's or non-perfusion areas or vascular malformation or CND. It's useful for disease classification. And we're also using generative AI to generate individualized normative reference for glaucoma and other optic neuropathies. And I'm sure that can be generalized to other diseases as well. And then, you can incorporate clinical information as well. I think there's a lot of interest in that. So almost every student or postdocs are using AI to some capacity in our research group now. And I certainly have had to learn a lot more about it in the past decade, kind of just through doing, since I'm too old to have learned that in school. So you are fortunate to learn it during your training years.
All right, let's join me to thank our speaker.